Welcome, everyone. Welcome to today's lecture on data safety monitoring boards. So I will, in today's lecture, go through a number of topics. First, I should introduce myself and some of the folks that have helped me with this presentation, but I'll say, there'll be time for questions at the end, so you can save them. But if there's something that is really important, please raise your hand and I'll make sure you get your question answered. So I'm an assistant professor at University of Pennsylvania, but before that I was actually here at NIH for several years at NIAID. And so I've done trials here at NIH, and then I'm involved in clinical trials, both on the investigative team and also on data safety monitoring boards. So we're talking a little bit about from my own personal experience and also just from the general wisdom about data safety monitoring. And I do want to acknowledge many people who contributed to this presentation over the years, including Laura Lee Johnson, who I think has taught a few other lectures here, Dennis Dixon, Michael Proshan, all either formerly or currently at NIH, and Susan Ellenberg at UPenn, who used to be at the FDA. Long list of experience, adding to a few of these slides today. So I'm going to give an introduction to this topic, and I'm going to just pretty much almost immediately launch into a couple of case studies or a couple of quick examples, because I think you really understand the what, the why, and the how by seeing examples for this particular topic. And then we'll cover the basics of data safety monitoring boards, how you form them, what they monitor, and the basic principles, when you need them and when you may not need them. And I'll finish with just some specific examples of things that are monitored by the DSMB, so going from the broad to the concrete through this. So the fundamental principle guiding data safety monitoring boards and overall concept of data safety monitoring is you can imagine you're doing a clinical trial of a treatment. It's novel, you don't know if it works, it might have some risks. You don't want to wait until the end of the trial to figure out whether or not during the trial something fundamentally went wrong with the conduct of the trial, essentially making the results unusable, or that the new drug had unexpected harms, or that there was convincing evidence very early on that you could have stopped and given this treatment to people much earlier than you did. Trials can go on for years. So ultimately, the idea is that clinical trials, it's an endeavor that's very important, but it's not without risks, and we want as many safety measures in place. And so data safety monitoring boards exist for that caution. Another fundamental concept is once you start, say, looking at data at interim looks during your trial, you can't just do that without any restrictions if you want a valid study at the end. And hopefully you've gone through some of these lectures on hypothesis testing, on randomization and maintaining the blind. Everything we know about clinical research really comes to a head in the concept of data safety monitoring boards. So one of the things that we learned from hypothesis testing, which I believe was already on your schedule, is that if you look at the data multiple times, you increase the risk of a false positive, right? So, you flip a coin with a 5% chance, eventually it's going to come up heads. And so if you think you need to look at data in interim looks because you want to make sure that if you need to stop the trial, you stop it, you need to have a statistical plan in place so that you do that and at the end still maintain the statistical validity in your analysis, that you want to maintain that acceptable rate, say 0.05 or whatever your desired rate is. It could be 0.01 for certain trials, that you maintain that no matter how many times you need to look at the data in the interim. And that's possible, but you need a data safety monitoring plan. Essentially, once you have interim looks planned, even by an independent body, you need a statistical plan that needs to be part of your protocol, generally preferable from the outset, from the design. And it also, you realize if the goal is to be looking at data in interim looks, then your data safety monitoring board is going to need people that can consume that data, that they're competent in understanding the difference between unlucky streaks in the data or statistically convincing trends, and that they can understand all the complexities of clinical research, including risk benefits and anything else that you might need to consider in order to make the decision of whether the patients are best served by continuing the trial or stopping the trial. And finally, if you have a group of people now charged perhaps at looking at data, or the only ones that may be unblinded, you need them to be objective, right? If they're going to be having this responsibility of deciding, is it safest for the patients, or is it best for it to continue or to stop, they can't have any vested interest in the trial continuing, right? Or perhaps some relationship with the study investigators or investment in the company. So suddenly you realize, based on the responsibilities of this board that initially we started with, well, we just want to protect patients, quite quickly becomes a very complex endeavor where you need to consider very carefully the people on the board and their responsibilities in order for them to be able to fulfill their responsibilities. So this definition comes, I think, after many years of trials, from their infancy, just people doing them, to having quite structured guidance from various bodies on when and how you create a data safety monitoring board. So the basic definition is that it's a group of independent experts that reviews the ongoing conduct and involving data, so emerging data of a clinical trial, to ensure continuing patient safety, as well as the validity and scientific merit of the trial. So not just that the data is not showing any early signals, but the trial that is being conducted is the trial that was planned, that the protocol is being faithfully followed. That's another sort of part that perhaps I haven't initially introduced, but I'm introducing now, that essentially that this trial is as intended and is ethical to continue. And this is just a brief note on terminology, because if you start reading, right away you start seeing that there are DSMCs and DSMBs, and mostly that's a cultural thing. Europe and the FDA tend to use committees, DSMCs, and that NIH uses the Bs, so Data Safety Monitoring Boards. I can just remember early on being confused if these were different types of bodies or are they the same body, but it's just general preference. But what's interesting is these groups are entrenched, and sometimes when they collaborate, they argue over what term to use. In any case, let's get into our case studies. So the first example, I think both of these studies that I'm going to talk about are historical, but incredibly exemplary in the sense of being the poster children for why you need a DSMB and the benefits of having a DSMB. So the first one is ACTG 076. So that's the AIDS Clinical Trials Group Protocol 076. And this is from the early days of HIV. And Sidovudine was just recently shown at the time to be able to slow progression of HIV. It was the first therapeutic breakthrough and the first treatment approved by the US government to treat HIV. So the trial on the docket that was going to be done was to see whether or not there was safety and efficacy in using Sidovudine, or AZT, in preventing transmission of HIV from infected mothers to their infants. And in particular, these women couldn't have advanced disease. And this trial, I give the results paper, which is the reference there. So at the time, NIAID had a group that basically ran all the trials that the NIH was launching funding for HIV, and they had a DSMB that was monitoring several of these trials. And so there were various ethical considerations from the get-go that they had to think about in designing this trial. And the first is, if partway through, they already have evidence that this works, that AZT works in general at slowing the advancement of HIV, there was a good chance that that could reduce infectivity of the mother. And so if you want to stop as early as possible, once you have convincing evidence, right, that you can prevent babies from being infected. This is a very grave diagnosis in the early 90s. So that's the first ethical principle that needed to be in the design of this trial. And the second was there was a lot of pressure to just give drugs immediately, but you didn't want to put out a drug that didn't actually work. And particularly for pregnant mothers and unborn infants or newborn infants, there are some extra risks there that we don't quite fully understand when we have new drugs given to pregnant mothers. So we wanted convincing evidence that there was overall benefit and not undue harm. So, that becomes a statistical analysis, and it becomes interim analyses that a body can look at during the trial. Basically, instead of waiting until the end, they're going to look at different times during the trial at these issues of what are the adverse events and what are the relative event rates of infection on those on a placebo versus an active arm. So it was powered to have an 80% chance to detect a 33% reduction of HIV transmission through the first 78 weeks of a baby's life. And so that was relative to a projected rate of 30% in what they knew about transmission at that time. So the target sample size was 748, and accrual began in 1991. So you might remember from the survival lecture at the end of last year, when you have time to events, time to transmission of HIV, babies getting infected, the sample size is the number of, typically the number of observed events that you need in order to determine the power. And so you have the idea of how many do you need to accrue to get the total number of events. So accrual began in 1991, and the projected accruals was to take at least 5 years. And actually, now that I'm reading this slide, remembering the 748 is the number of mother-baby pairs you needed for the expected event rate to have 80% power. So already you have to do a little bit of a guessing game for sample size, which is another reason why you might have a DSMB look partway through to make sure you're on track in terms of your projections and having adequate power. So the projected accrual was to take 5 years, and they were planning for 15% dropout. So the monitoring plan in place was to meet twice a year just to look at safety. Automatically, that was happening. And in addition, they were going to do efficacy reviews, and that was after each third of the projected infant infections. And then they projected that the first efficacy review should take place in order to capture about a third of the events around February of 1994. And so then that would happen, this is a little bit of the nitty-gritty. You can't just say, okay, we'll use all the data up through January 31st, right? So they basically locked the data for all mothers enrolled up to December of 1993, and then they would look at the events up to that point. So this is the data analysis at the first look. And so what we're looking at here is sort of the opposite of a Kaplan-Meier curve. So normally survival curves, you look at the event rates. Nobody has the event, say, mortality at the beginning of the trial, and then they decrease, and then you can compare the mortality rates over time or the percent alive. Here what we have is, some babies might be born basically instantly infected, and so you'll see that the event rate, let's get this going here, is not quite at 0. Maybe it's a little bit higher. So babies are essentially born already. There seemed to be an apparent difference. And as time went on, the zidovudine arm only had 8% of the babies infected, whereas placebo was projecting 25% by the time a baby got to 72 weeks. That's a more than fourfold difference. And the p-value was really convincing. And at this point, if you look at the total number enrolled, it was about 360. And I think basically they had about half of the planned events that they were expecting. And so quite a lot of information had accrued. Events and accrual had happened a little faster than expected. And so this was overwhelming evidence and certainly met some conditions where you might think, well, okay, we definitely need to stop. We have evidence of benefit. But there were some discussions. And so the first discussion was that they knew there were 46 babies that were sort of in limbo. They didn't know their HIV status, and they didn't want to stop for efficacy and then have those babies evaluated and realize retrospectively that the boundary wasn't actually met for stopping. And so because there is more evidence that's needed, not the usual 1.96, and we'll get into that in a little bit, when you're stopping early. And so there was some statistical analysis to look at reasonable trends of what could happen to those 46 and what could happen to the p-value depending on different outcomes. That was considered. They looked very carefully at safety. And that type of analysis that I'm talking about, just to explain all the words on the slide, is what we call conditional power. So conditional power is essentially, given the data up until this point, what's the probability that you will wind up with a positive result at the end? And so that is often a statistical analysis that will be presented to the DSMB when pertinent, when you're starting to think about, is it time to stop? So after careful review of that conditional power, of the safety, of how much data was accrued, which was quite a lot, the DSMB did recommend that there was overwhelming and convincing statistical evidence that there was benefit to this treatment, and they recommended the trial stop. And a key term here is the DSMB is a board that's an independent body. They're not the sponsor, they're not the investigators. They give advice, guidance. But in the end, it's the trial leaders and ultimately the sponsor that will make the decision, typically on whether the trial should go or no go. And in this case, as in many cases, they followed the advice of the DSMB, the recommendation, and they stopped. And instantly, this zidovudine was provided to those in the control group, and guidance in the US and across the world was changed to allow this to be a treatment for prevention. So that was a great success, and the benefit of having the DSMB was that they didn't have to wait to the trial's end. They got to stop actually a couple of years earlier and get that treatment out. So many babies were able to escape infection because of that earlier stopping and distribution of the treatment. So here's a different kind of example, and exemplary in another way, and it's CAST. And there's actually many, many lessons that can be learned from this trial, as is typical when you do a large trial. So this was the cardiac arrhythmic suppression trial, and it was designed to evaluate a hypothesis of whether suppressing ventricular arrhythmias in patients with recent MI or heart attacks, whether or not that would reduce sudden death, that was the primary endpoint, or overall mortality as a secondary endpoint. And there were actually several active drugs that were known to be successful in suppressing arrhythmias, and so this trial looked at 3 of them, econide, flecainide, and morosiazine, which a little more shaky about how to pronounce that one. You can see it on the slide. So we had 3 actives and 3 matching placebos, essentially a large, complex trial. And they were randomized 4,000 patients, partially because they were studying several treatments at once, also partially because they need for that event rate. There's a number of patients you randomize, and then not all of them will wind up with those events. And as we learned, it's the number of events that's your true sample size. So they needed to randomize 4,000 for 90% power in the end to detect a reduction in sudden death with a 0.05 one-tailed significance level. So that was the original design. And I don't know if any alarm bells are going off, how well Laura Lee jumped up and down about this. It was Laura Lee Johnson who did the hypothesis testing, yes. But so the DSMB met for the first time. So this cardiovascular trial was launched in 1987. The first meeting of the DSMB, so this was a blinded trial, large trial, death as an endpoint. It's going to have an ethical monitoring board as an independent. Phase 3, basically required to have a DSMB. Their first review was before even the study started, and that's often the case because they want to look at the protocol. They want to make sure that this monitoring board is okay with the protocol as is and agree on their charter. What are they going to monitor? Is everyone on the same page about what's appropriate to monitor and how? And as a result of that meeting, they made a recommendation, actually, to change the primary analysis of the trial. And actually, 1987 now is going on 30 years ago. And statistics was getting, in clinical trials, was still sort of being formalized in terms of what are best practices. And perhaps this is a time when it basically became convincing, and certainly there was a forward-thinking statistician on that DSMB that just was no longer acceptable to use one-sided 0.05 level tests. So the initial argument is we know the smaller the alpha, the more difficult it is, the more N you need to guarantee that low of a false positive rate. And someone might argue, why would I want to spend alpha to look in either a strong positive direction or a strong negative direction to decide whether or not there's a treatment difference when I'm only interested in the strong positive direction of benefit? I want all my alpha going to the 0.05, and then I can use 1.64 and not 1.96. But the opposing argument that has become the dominant one is that you don't want a test, whether it's one-tailed or two-tailed, to influence the level of evidence that you're going to use to declare benefit. Right? So whether you can do a one-tailed test, but it shouldn't be easier to pass that test than a two-tailed test in terms of deciding what is the benchmark, what is the critical value, in this case 1.96, to declare significance in terms of a difference between two groups. Say if you're doing a one look at a 0.05 level. And another argument is that, you almost rarely, I've never seen a one-sided confidence interval. When you do a confidence interval, you do the uncertainty, plus or minus, say 1.96 standard deviations around the mean. If you do your test as a one-sided test and a confidence interval as a two-sided confidence interval, you could wind up doing this test, declaring a difference, having a confidence interval, and having the null inside of that confidence interval. So it's, some might view, if you're more on the clinical side, this is like an esoteric argument. Why aren't the statisticians on board with this? Well, they are now, but at the time, perhaps it was an emerging consensus. And it was interesting that at the very first meeting, essentially the principal analysis was changed. Well, they made the recommendation, and then the study team and the sponsor were convinced by the arguments, and they did change it. And it only reduced the power a little bit. So they were already going for 90% power, which is quite convincing. So they were still at 85% power when they did this change to be two-sided. So I think in the end, everyone was satisfied with that change in the protocol. And so the lesson learned here is it is very important to have that first meeting before the study starts. Because ultimately the DSMB is charged with a pretty delicate sense of responsibility, actually a very important role, which is they are going to be looking at data that no one else has and be making recommendations that have a strong influence on the future of this trial. Everyone needs to be on the same page scientifically, believe in the trial, believe in its validity from the start, and then be able to trust each other and get along through what could be potentially difficult times in terms of unexpected challenges that make this trial more difficult to complete than anticipated. So you really want that first meeting so everyone is on the same page about the roles and responsibilities and the science. Another lesson learned was not only should you be on the same page about the design, but the monitoring plan should be in place. Because what happened at CAST, which I think was just one of those every 6 months, basically there are two kinds of meetings the DSMB has. One is just regularly scheduled, we'll just keep an eye on safety, and then you'll do these interim looks at efficacy as you go, not expecting real efficacy trends until a lot of data has accumulated. But in this trial, with only 5% of the expected information, 5% of the total number of events that they needed for that power, they actually already had a difference apparent between the treatment arms. Now, as is typical in a DSMB report, once you start looking at efficacy, the report is going to just have arm differences, and you might have them labeled as arm 1 and arm 2. Because you never know, you want to be, out of a preponderance of caution, you want that report to be masked in case it gets into the wrong hands, mailed to the wrong person accidentally, left on a desk. But then the DSMB would have the potential to say, okay, tell me which arm is which because I want to make a decision. And others will always have that which is which, say in a separate envelope that they open up at the start of every meeting. In this particular trial, as was done at this time, more commonly than it is now, they chose to remain blinded until perhaps they decided there was a reason to look. And so they looked, all they could see was there was a treatment difference. At the time, they hadn't had stopping boundaries in place, perhaps because it was so early and they were still gelling the analysis plans. And so in the end, it was probably fine because even though there was a convincing, when they did the test, in this case it's a log rank test because they're looking at survival curves between inactive and inactive, or say placebo, the log rank looked like it was exceeding. It was 3.43 for a treatment difference, and the boundary that they were eventually applying, retrospectively looking back, it seemed that they had exceeded it. But it's that issue of there were pending events, and actually when they were adjudicated and added in, it actually would have flipped it so that it would have been less convincing. So in the end, even if they had the boundary in place, even if they had looked to see which arm is which, they may not have stopped. But it was instantly alarming. But they chose to remain blinded. And at the next look, and there's a reference there for this trial that people talk endlessly about for many reasons. At the next look, there were 48 events. So at the first look, it was roughly 22. now there were 48 events. And actually looking at event rates and other reasons, they realized that they would only expect 300 and not the 400 and some they'd originally thought at the end of this trial, which meant that 48 was a little bit higher of a percent in terms of total number of events. And when they looked at the boundary, they saw that their log rank was actually still at 3.22, and it was well over the boundary for stopping, plus or minus 2.98. And so the DSMB requested to be unblinded to find out which arm was which. And much to their surprise, it was the placebo that was doing the best. And it was active arms that were having harm. And all along, everyone had assumed it was benefit, that just they were waiting for it to be convincing. And this becomes the compelling argument for why most people argue you should not say, well, to remain objective, I don't want to know which arm is which until I really think there's a difference. And the reason is you're probably not going to make a symmetric, even decision in terms of how much evidence do you need to convince the medical community to adopt this new therapy, which might have harms as well as benefits, versus evidence say, you know what, I'm 80% confident that we might be hiring people, but I think we should wait till 90% confident because it's kind of early. No, so it's really not a symmetric operation. And I think having seen this over and over time, people have just, a lot of the think tanks that make recommendations on how to do these things, people who have served on these bodies many times in their careers, make an overwhelming recommendation that the DSMB is not masked. Trial is double-blinded, the investigators and the patients. It is not triple-blinded. We do not blind the DSMB. We want the DSMB to have every bit of information possible to make the decisions. And this is ultimately why. Because you can't help but sort of believe in the new treatment. And in fact, they were so convinced that something had to be wrong that they investigated, they got random bottles. They assumed maybe the place where they were labeling the bottles had made a mistake, and that the placebo was the active and the active was the placebo. So they started testing chemically bottled medications that had those labels. They looked for imbalances in key variables in the placebo and the active arms to say maybe the differences were because these folks weren't actually have the same risk factors. And then they continued to also look to see if there were certain subgroups that maybe were being harmed versus others, again thinking, well, if that subgroup was out of balance, that might have driven this treatment difference. And they also looked at the secondary endpoint, which was overall mortality, to see if it was consistent. And finally, when they realized it was in fact the placebo that was doing better, they just wanted to see whether certain arrhythmia drugs out of the 3 that they were studying that were actually the problem. And it turned out early on that there were 2 out of the 3 active drugs, echinide and flecainide, luckily the 2 I can pronounce, that were driving the difference. So each of the 3 actives had a matched control, and so the overall score was 35 events on the active, 13 on the placebo, and when they reduced it down to those on the echinide and flecainide versus placebo, it was 33 versus 9. So what had just been CAST became divided up into CAST 1, which was the trial with all 3 treatments, and then at the end of this DSMB, they stopped the echinide and flecainide arms, but they continued the third active and matched placebo because there was evidence, certainly there was no reason to stop those arms. And so that was the benefit of having sort of the structure of multiple matched placebos, that they can continue the one question even though they had to stop the other 2. So other lessons learned here is that not only should your monitoring plan be in place, your DSMB be in place all before your study starts, but your data management system, your data entry, all those people in charge of monitoring your data, entering your data, organizing your data should be on board on day 1, and that data system has to have been built and tested because you need that data at that first meeting if it's available, because very early on trends can become apparent, certainly for harm. And also, it turns out, for benefit, say in the AZD trial. And other principles are, you need to be organized, you need to have these meetings on the books, because there could be many reasons why you need to act quickly, so start your monitoring right away. And also, you need a board that's going to be able to think about, and investigators, contingency plans about what to do once you have this information. So people need to be ready to receive the DSMB recommendations as well. And I think, as I mentioned earlier, I think probably went over most of this, is that the ability of the DSMB to do their job can be compromised if they remain masked. And I think initially that idea of objectivity seemed to be the overwhelming principle of why we would keep the DSMB masked, unless, and perhaps always give them the prerogative to ask to be unblinded. But I think it was, examples like this, that in general, I think the recommendation is that you keep a DSMB body unblinded. The report may stay masked because you never know who could intercept it, but you would always have that DSMB have the ability to see which arm is which. And a great article, and it's just an editorial, it's only about 2 pages, that really summarizes both sides, I think, but it's definitely by the name of this article, Mass Monitoring Clinical Trials, Blind Stupidity, ? Obviously coming down on one side of the debate here, and certainly a good way to augment reading if you're interested in this topic. Because they do present the opposing view, at least briefly. Okay, so hopefully those 2 examples, sort of have given you some ideas of why we need a data safety monitoring board. Essentially, to summarize those examples, we need to be able to identify any safety problem rapidly. We need to be able to identify logistical problems, if there are some, so that the board can recommend amendments or make sure that those come to light, such as, if the accrual is so slow, they'll never get full enrollment by the end of the grant period. That could be a problem, so maybe you need to add sites. So they'll be actually evaluating the feasibility of the trial, not just things like efficacy or safety. And ultimately, they want to make sure if the trial objectives have been met early, then the trial should be terminated early. However those objectives may be defined. And so the things that you would be monitoring to accomplish this, you can imagine, so are you doing the trial you intended and is it feasible? You're going to monitor various logistics like enrollments, baseline data, comparability of the arms, protocol compliance. A lot of times people have visit windows that are impossible. Like, everyone should come in plus or minus 1 day of their anniversary. Like, when you wind up with 90% of your data in violation of the protocol, you rethink the protocol. Specimen collection, is it being accomplished? Are your data complete? And is the quality in terms of, generally these databases are independently monitored from the sense of a data monitor comparing source documentation with what's actually in the database, and those kinds of statistics would also potentially be reported to a DSMB. And ultimately, so you would agree on, say, these will be the logistics that the DSMB will keep an eye on, and the sponsor participates in that conversation of what they want to be monitored or considered by the DSMB. And it's generally best to think in advance about specific benchmarks that you'll start to think about will be concerning. Obviously, you'll be monitoring outcomes such as adverse events, interim variables. If this is a trial for vaccine efficacy, and it might be a while, say if it's HIV prevention, it might be a while to get trial results. You can certainly look at the immunogenicity. You might expect if this vaccine is working that certain immune markers should be elevated, right? So if early on, it's all flatlined, and you're also getting evidence of very similar event rates in the groups, and there's no evidence that there's immunogenicity of this vaccine, and this could be something that would give early signs that there could be an issue with futility. And finally, if agreed in advance, you might be also actually monitoring your primary endpoint. Again, to see if you can stop early, and the patients are best served. If you can answer your question more quickly, and convincingly, then do that. So, who monitors a trial? Data safety monitoring is a very generic term in some sense. You're going to have many ethical committees next week. You'll hear about IRBs or one of the next lectures. The sponsor is going to want to keep an eye on things, say from a general perspective of accrual, or certain serious adverse events are always reported to the FDA, to the sponsor, to the investigator if they're serious. So many folks are are looking at, there's a safety monitor, they're looking at the patient safety from different perspectives. But I would say it is unique responsibility of the Data and Safety Monitoring Board to have that that goal of looking at potentially at the efficacy. In fact, they're the only ones that should be looking at efficacy in a blinded trial. Otherwise, it's not a blinded trial. And so they have basically the unique responsibility of being able to look at the totality of evidence. And that's what makes them so important. So remember, their purpose is to look at that regular interim looks at the data. They need to provide an objective assessment of whether or not they're on track to continue or whether there's reasons to stop. And they need to keep everything that they've learned completely confidential and out of the hands of the sponsor and out of the hands of the investigator, because if the idea is the trial should continue as planned as a blinded trial with equipoise, then nothing should be leaked. And that sometimes is difficult to accomplish. So, generally accepted that, think about if those are the goals, when you're forming these DSMBs, you realize they should be multidisciplinary in order to sort of be that scientific guide, that statistical guide, that ethical guide. They need to also have a well-defined charter, set of roles and responsibilities. They need to be free of conflicts of interest, and they need to be people that understand and protect confidentiality. And I think just it's so important, I want to just emphasize, why confidentiality is so important, why they can't just leak some kind of summary of how the trial is doing so far with efficacy, and that goes back to why we blind trials, right? So if you had some idea of how this treatment was doing, it may affect what kinds of patients you enroll. It may affect the care of the patient, whether you think they're suddenly more at risk or doing well, or how you assess patients. Maybe you're hunting around for a certain kind of response, and it might make a sponsor nervous. And these could all be just streaks in the data that weren't statistically convincing at all. You don't want any of that spuriously influencing how we conduct the trial, which hopefully is completely objectively just randomizing patients to different arms and evaluating them all the same way to get a reliable answer at the end. So if partial knowledge of interim data gets out to people that are enrolling patients or recording data, I think it's generally believed it could muddy the waters. And so really no interim efficacy should be revealed. And I think that's why there's so much emphasis on independence. So not only to maintain objectivity, but you can imagine if a member of the DSMB is the best friend of the protocol investigator or a spouse or of someone involved in the trial. Our research worlds are quite small. It could be almost impossible to, I think, be immune to that pressure or to just have it be in situations that are unexpectedly compromising. And so for that reason alone, having people that don't have contact regularly with the protocol team is an easy way to remain, basically a firm boundary between those protected information and those that should not have that information. And generally also just to maintain that as you'll see, if you read more case examples, that this data can get very murky. And so you don't want anything to influence you other than the data itself, in terms of being invested for the result to go one way or the other. So you consider that when you establish a committee. It is generally appointed by the sponsor. They want this study to be a success. They're going to be looking for clinicians of the appropriate specialty. They'll be looking for statisticians, without exception if there's going to be monitoring boundaries and statistically technical analyses to be evaluated. You need people to consume that data and people to present that data. There could be ethicists, particularly in trials of vulnerable populations. There could be subject advocates. And there's always an executive secretary in the end who makes sure that this meeting is run, that the charter is followed, that minutes are taken. It's a very formal responsibility. And membership needs to be acceptable to the trial leadership because they have to trust this board. And a fundamental principle is that no one on the protocol team is a member, or certainly not a voting member, of the DSMB. Interestingly enough, there are differences of opinion on whether or not the study sponsor can be on the DSMB. There's guidance from the FDA that says sponsors should not be part of the DSMB. There should be no member from the study sponsor on the DSMB. However, at NIH, the perspective at many institutes, not all of them, but at many institutes, is that they serve the public. They're not financially invested in the sense of profiting from these trials individually. It's a not-for-profit research organization. And so the public is best served if they are members. They are the experts in these fields and in running these trials. And they're an advocate for the public. So what's interesting is something as integral, as important as the DSMB, there's actually still quite a bit of wrangling over the best way to do these things. And there's probably not a one-size-fits-all. I think that's something everyone can agree on. The meetings generally have a recommended structure or a typical structure in any case, that you would have an open session where you process the data. The sponsor, the investigators are summarizing the trial, the accrual, and some unprotected information about how things are going. There are often site representatives there, and they're discussing data quality. And then there will be a closed session in which the investigative team, the protocol team, people running the trial would all leave. And that would be when you would essentially be presenting data by arm, certainly any unblinded data. Certain types of AEs are indicative of that, that belongs to certain kinds of treatments. So if you knew that AE rate, you'd understand how people are doing on a particular arm. Those kinds of data tables would be reserved for the closed session. There typically needs to be someone who has access to the study data and the unblinded study data. That is often going to have to be a statistician that somehow is there sort of at NIH or at the group that's doing the trial. And so there's various ways that you have an independent statistician that's associated with the study, but at the same time has no contact with the study team to maintain that confidentiality and blindedness of the study investigators. And you can see this is quite a complex endeavor, almost a little bit of cloak and daggers. And then finally, there'll be an executive session. So generally, you do need a data expert that is in some way affiliated with that sponsor and that study. But after they present the data, they're done. Oftentimes it's a statistician who they believe, even when they belong to a group, sort of Spock-like in their lack of preference for how things go, and they only care about the numbers. But they're finally asked to leave as well. And then the executive session should only be the DSMB members that vote, that are truly what we consider those objective members. And then they discuss the totality of evidence and any recommendations that they feel they need to make about this trial, which is to continue as is, or if they feel there are modifications or reasons to stop. There can be special requests to have someone come back in to discuss an item. Obviously, this is in some ways there's structure, in other ways there's unstructure, in the sense that every trial can present a different challenge. And so the scope of the responsibilities for the DSMB is that they're going to evaluate this accumulating data with respect to toxicity, right? They're going to think about potential efficacy and or futility. They'll be looking at risk benefits or potentially the complete lack of expectation that there could be any positive result at the end. So they can recommend, after that totality of evidence, terminating or continuing. If they're concerned about accrual or other issues, potentially a certain AE that they're concerned about, they might want to see more data. And how they make those requests can sometimes give signals, so they might only make those requests to that independent statistician and not to the team as a whole. Even that has a little bit of structure that's delicate. And finally, they might ask either for the next time, if they don't think it's really a hurry, something that would be more helpful next time, but they might ask for additional analyses than what was initially anticipated. They basically have full reign to ask for what they need to do their job. And a key thing, though, regardless of what they ask, is to think about the statistical implications of that. So certainly, either if it was planned from the start or something happens where they add this as a modification, once you start looking at efficacy, that's inflating your type 1 error if you don't adjust the statistical analysis for those added looks, essentially requiring tighter evidence each time. More evidence early on, and certainly still more evidence at the end because you've looked multiple times. And there are many methods to do that, and so it's important that you agree, certainly as soon as you realize you're going to need that plan, on what plan would be used to make those decisions. What stopping boundaries would be consulted, right, in terms of essentially there's a continuous boundary in terms of amount of information collected so far, test statistic at that time, and whether or not you're above that level or below that level would be the reason to stop. And just to convince you, or just perhaps to make it more intuitive, as to how quickly additional looks can inflate your false positive rate. So that false positive, the idea that there'd be two arms where there's no difference at all. But if you keep looking, maybe you have more chances that just random streak could create a difference. And so on the first column here, we have the number of looks. And say 1 would be a trial with no monitoring. At the end, you just look and you do a test at 0.05 level. At the extreme, you do 10 looks. If you did 10 looks, say equally spaced, and you always use 0.05 to decide at each of those 10 looks whether or not you would stop in terms of the difference, the p-value was less than 0.05, at any of those 10 looks you'd stop, you can see that actually your error rate would be 19, almost 20% instead of 0.05. If you did 5 looks, your error rate would be almost 15%. So it does increase quite quickly. So even if you're doing 2 looks, I think you should adjust. And that's just if you're doing equally spaced. There's some ways you could game the system in terms of doing the looks when trends appear. This is a little bit mathematical, so I'm not going to explain exactly how I define this worst-case spacing. Let's just say there are ways to game the system and the timing of the looks so that the type 1 error rate could be much larger, as high as 45%. And it sounds ridiculous, but I've certainly been involved in, not in trials, but in analyses, say, with investigators, and these are mice, which are a little bit easier to recruit, say, to your trial, that you do an analysis and you realize, the p-value is 0.06. Let's add another mouse and not quite there yet. Let's add 5 more mice. Hey, now it looks kind of convincing. Let's publish. Right? There's something inherently wrong with that. Let's add mice until we get a difference. You need to adjust the 0.05 level. And so that is an example of worst case, or getting close to worst case. That's not equally spaced at all. Okay, so once you set up these boundaries, which are going to be, I'm not going to use the 1.96. I'm going to make it harder at the beginning. Maybe I'm going to need a 3. There's some mathematical principle that's going to guide us. The thing that's interesting is that if you read examples of these trials, you can kind of see what random streaks do. But there's all kinds of examples where you get incredibly close to the boundary and you're all ready to stop, and in the end, a completely opposite decision is made, which emphasizes, I think, just the need to have the structure, because I think randomness is a little counterintuitive. Like, we think equally spaced is somehow random when actually that's quite regular and hard to achieve. So the Coronary Drug Project was one of these large cardiovascular trials. And once they set up the boundaries, they actually reached minus 1.96 3 times, which isn't reason to stop because they needed stricter rules because they were looking multiple times. And in the end, they wound up at 0, no difference. There was another trial in the literature that's historical. It was called MUST. And this was looking at whether or not if you did guided treatment of arrhythmias with this electrophysical testing, whether that would be a benefit or not to the usual unguided treatment. And that appeared harmful right away and stayed that way for quite a bit, and then in the end actually proved to be beneficial. So I think it's these sorts of examples emphasize the need of a sort of interdisciplinary body that can really think about the pluses and minuses or the risk-benefit to the patient, and also, if you stop too early, you're not going to convince anyone that the treatment was harmful or beneficial. That's the worst-case scenario, that you have to start all over again. And there are examples of that, where in the end, people were convinced neither way and were still interested in the treatment, and they had to start all over. And to give you an idea of how these boundaries work, and actually that there is something to choose, although I think there's one common one that people typically do. But so you think about this is the z-score boundary. If we did one test at the end, we'd be down at 1.96. If you do lots of tests, think about here on the x-axis, we would have an interim analysis. Maybe we do 4 looks. So you can think this is in time our looks. And you want to make it, the O'Brien-Fleming principles, you want to make it really hard to stop early on because you want to be swayed by those streaks in the data unless they're really convincing. And then that way you also benefit by having this sort of steepest descent. You wind up with the smallest possible penalty at the end where it's not 1.96, but it's just a little bit above 2. So you have some chance of stopping early, but maybe not a ton. Hey Biddle Pedo and the Cat in the Moon. They say we're going to do this really simple. P less than 0.001. If you're less than that at look 1 or 2 or 3, you stop. That's actually 3 times that is 0.003. That's barely an inflation if you think about it. I could make my p-value at the end 0.047. And so I do 3 sort of even amounts of evidence across these different looks, and then at the end I only have a tiny adjustment again, which is thought is desirable because the bigger the adjustment, the more people you need for that convincing trial at the end. And I say that there's a lot to be said for simplicity. If there's not a lot of staff, not a lot of experience in the statistical team, everyone can implement this boundary. The negative is, basically make it hard, and then kind of relatively not so hard at the end, is that you could have had evidence here that was towards a benefit, but not quite at that 0.001 level. And then you could have had all of the data accumulating that was in the wrong direction and still wind up less than 0.05, somewhere between 0.00, say 2, and 0.05. And there's something logically inconsistent about that. That you wouldn't have stopped for benefit at a p-value of 0.002. Then you had a bunch of a negative trend accrue and then stop for benefit. And so mathematicians sort of took a hold of this and tried to find a compromise that would achieve these goals in a way that would be logically consistent so that you couldn't somehow get stronger evidence by reverse trend. Pocock wanted it to be easy to stop early on, concerned about safety, but that boundary has similar problems with the pedo and is actually quite an expensive penalty in terms of needing a lot more people in the end, and I think he actually recommended against his own boundary. It seems strange to have the same, well, we'll leave it at that. The overwhelming, I think, particularly for large phase 3 studies, the overwhelming way that people do it is they find this thing they call alpha spending, where you consistently sort of spend some of your alpha, your 0.05, that's your allowable false positive rate, across these looks that for this boundary, magically, you don't even need to say how many looks or when the looks are happening. And you can have different mathematical functions and achieve this spending function, which basically spends the alpha, think about it in the steepest way, the fastest way, and still wind up spending 0.05 at the end of the trial, or spending it the slowest in terms of it spending very little alpha. We've got sort of 0 to 0.05 on the y-axis here, and then spending it all at the end towards the final analysis. That's the O'Brien-Fleming spirit, but done in a way that we don't have to specify the number of looks, but instead always just adjust for the percent of data in versus the percent of data that's left. And these 2 statisticians, Gordon Land and Dave Demetz, came up with this clever way of doing the monitoring boundaries. And so it's become a pretty standard way to do trials. You still see the hey biddle pedo because of its simplicity, maybe in early phase trials that have less of a budget for lots of staff and fancy statisticians. But in the end, there's software now that just about anybody can do these more technical boundaries. You just need to do the reading so you understand how to do them correctly. Okay, so we've talked a bit about reasons to stop. You achieve your goals, you get convincing evidence early, you have safety signals. Another area that DSMB monitors, often at the request of the sponsor, is if it's futile to continue in the sense that the trend is overwhelmingly negative, no harm, no benefit. You've got 4,000 people lined up in the randomization. Are you going to wait how many years are you going to wait for a null result? If you can tell partway through that the data is so incredibly null that even if you accrue the next 500 patients, it can't reverse the null, You will never get below that stopping boundary or that significance at the end. That saves expense. It saves patients for more promising treatments in future trials. It frees up investigator time to go after those new other developments that would be more beneficial. So there's many reasons why you would actually want to think about what's called a conditional power analysis, which is given the trend so far, what's the likelihood you would have a positive result at the end given the planned analysis. And so that's one way of being futile. Other reasons might be you can just look at the recruitment rate and the writings on the wall. This grant ends in a year. Look, you're not getting 200 people from this rare disease. Event rates might be too low. There could be unexpected dropout or crossover. Sometimes you're studying something that's easily available, like screening. And so you might realize that you just can't achieve the scientific goal. The one area that's different than the rest, or what I've listed here, is poorer than expected treatment effect on the primary endpoint. So that's slightly different, or at least I'm trying to phrase it so it's apparently different. If you want to prove that something is poorer than expected, poorer than you would hope for, then that's an unconditional power. Like if you power the trial to have 90% power to detect at least a 25% reduction, if you fully enroll and don't achieve that, then you have you sort of 90% confidence in the sense that the treatment effect is lower than what you would power to detect. And that might be useful. For instance, a lot of people take glucosamine because they think it helps their joint pain, or they take St. John's wort because it helps, and they think it helps depression, or other treatments that might not be so innocuous, and you might want to convince the community to stop doing that these treatments might have, certainly expense, potentially harms, and false hopes. And so you might want to finish a trial even though you know it's going to be null, because you want that convincing null result, a meaningful null result, that you had the statistical power to have detected something greater than 25% reduction, and you didn't see it, so you know that with a good deal of certainty it's not there. That's an actual argument to continue the trial, as long as there's no safety compelling you to stop early. So the fact that you might be heading towards a null, that might be useful and not necessarily a reason to stop. It would really depend on the circumstances. Typically, if that drug isn't on the market yet, and then it has no chance of approval unless you hit a target of efficacy, then you do futility monitoring for the purposes of stopping early and dropping that treatment. But if it's already in use and you want to convince the medical community, then I think that's sort of more on the scale of you wouldn't stop a trial for futility, or you have secondary endpoints that are just as important. A lot of times a trial has multiple goals, and as long as there's not a safety problem, you might not even monitor for futility of the primary if you know you're going to achieve something useful out of other endpoints. Okay, I think I've already covered that slide. So, this futility monitoring can be summarized by, you might want to ask two different possible questions. Will in the end the result be null? Will in the end the result be null and still meaningful? The first one is when you want to stop early. If you just, you might do a conditional power analysis. What's the probability of getting a null result, given the data that we have so far? And the other one, if you want a meaningful, interpretable null result that gives you information that the efficacy is lower, then you need the fully enroll, and you would rely on that unconditional power. Okay, so summarizing, monitoring recommendations. Once you look at the totality of evidence, efficacy, futility, safety, study validity, the board would think about making recommendations to continue the protocol unmodified, to continue but modify, potentially to terminate, and they summarize those and say a set of bullet points that they give to the investigators and the sponsor. The sponsor, in the end, makes the final decisions to adopt any of those recommendations, and certainly whether or not to terminate, as we've talked to before. Here at NIH, it's the institute director that makes the final decision. And in general, we'll follow the recommendations of the DSMB. There are interesting examples in the Women's Health Initiative and the hormone replacement trials. Is a fascinating one to look into. I think they disbanded the first DSMB because they didn't like the recommendations. They wanted an independent second opinion. And you can imagine how that first DSMB felt. And so I would say this is, I mean, if you like suspense novels, I would reach for a case study of DSMBs because they can be cliffhangers and with lots of twists and turns. So ultimately, a DSMB could recommend stopping, which is the hugest possible outcome of a meeting. And that could be if the safety issue emerged, if efficacy was achieved, that's best case scenario. Interim results preclude a positive result, that's the futility. This is a disappointing one, that operational difficulties are insurmountable, generally because either the primary endpoint was just infeasible and could not be obtained in enough people without and too much missing data, or accrual was just in the pits, and you're just not going to be able to add enough sites to finish on time. Interestingly, sometimes when there's multiple countries, multiple groups could be studying similar therapies. There could be other trials that provide information that give you information about what you're studying, either is no longer being needed or potentially sometimes you can get safety signals from other trials that were slightly ahead of you. And so that's important when you're on DSMBs to be aware of people who are other countries or other groups that are studying similar treatments. And oftentimes those DSMBs are formally put in contact with each other. And I think, hopefully, as it's just this lecture always seems to fly by. There's so many issues to talk about, and I always feel like I didn't get them all in that I wanted to. But hopefully, you can appreciate, at least from the brief examples we've talked about, that the decision process of a DSMB is very complex. You have to worry about internal consistency, that this data is meeting expectations, or is consistent with the pilots going in. You think about external consistency. There might be other groups with related results. You're thinking about benefit and risk, the balance of the patients in your trial, who are ultimately only a small number compared to the total number of patients that they're representing, and how well do they represent those people in terms of their covariates being what you had hoped as being a representative group. And you think about that clinical and public health impact of having convincing evidence, not only that this treatment works, but to make people change their practice. And that can be quite an amount of additional evidence. And then, of course, the statistical issues of sorting out signal from the noise can be quite complex. And especially when you have rare side effects that are extremely worrisome, you don't want to wait until you have statistical evidence to prove without a doubt that you're harming people. And yet you also don't want to be swayed by one or two unusual events that might have been due to chance. And there's just no hard and fast rules to be able to sort that out. So it's a lot of hand-wringing. So I hope that really gets you interested in joining one of these DSMBs. They're incredibly important. They're voluntary efforts, and really it's about protecting the patients in the trial as a first responsibility. And the one thing that is also wanted to emphasize in terms of the complex decision-making of this body is you have that individual versus collective ethics, which perhaps you've already started that discussion in this class and will continue as you talk about IRBs, I think. That as you decide whether or not the patients are being best served, you're thinking both about, primarily you should be thinking about the people that are currently taking this drug and being exposed to risks, but you're also thinking about their future and the future of other patients as you make your decisions. And that's somehow having to work in that collective ethics. And on top of all the technical complexities of data and noise and signal, you have what can be complex cultural and ethical decisions to sort through, and something that recommending stopping should not be taken lightly for many reasons. And one of them is actually that stopping early handicaps safety analyses, right? You're not getting the full amount of data that you could have had about this drug. It can actually affect it. They have shown statistically, essentially there's a little bit of bias in your treatment effect if you're stopping early for benefit as well. And people have fixes for that, but that's a little bit more esoteric. I think the more complex one is that clinical trials are very good at comparing the arms of the people in the trial, and they're less good about making more absolute statements. And so sometimes a trial can end, such as the Women's Health Initiative. The hormone replacement trial, we learned women who basically started hormone therapy perhaps some years after going into menopause, had excess stroke and excess, certainly no benefit in preventing breast cancer. And yet that raised, and of course, women immediately stopped taking these drugs. But more questions have arisen about, was that the right population to study? And well, many women would have started those drugs much earlier. Are the risks the same? And so sometimes stopping a trial is not necessarily the end of a question. And I did want to just finish with one last example before getting into some of the sort of nuts and bolts of the nitty-gritty of the meetings. And this has to do with that idea of, it's not just a statistical decision. You know, the stopping boundaries, and a perfect example of that is well, I would have said recent, but I guess it's already going on 10 years. But this was a collection of trials that were done in Africa in terms of looking at whether or not male circumcision would prevent HIV acquisition. And so there were phase 3 trials, large trials, that began at the same time in South Africa, Kenya, and Uganda. Typically what they're doing is they're taking a group of people that they're recommending, and this is not a typical practice. And so there's a group of people that would be randomized to get usual care, and another group of people that are going to be randomized to be heavily counseled to take on circumcision, and you're randomized to basically say you would accept either outcome, so that you don't have a bunch of people that sort of got the wrong arm and quit. But in any case, they had several trials that they had going on in Africa, South Africa, Kenya, and Uganda. The designs were very similar, and this is a cultural practice, and so there was thought you would need to study it in multiple cultural settings in order to get an idea that this was going to be working in each of those settings. So the South African trial had a head start, and early on, they had clear evidence of efficacy, and they stopped early. But I think the DSMB was quite sensitive to the fact that folks in Uganda may not accept a result about South Africans or think that it pertained to them, and there could be real cultural and behavioral reasons why it might not pertain to them. And so for many of those reasons, the DSMB decided to continue, even though they had evidence that in theory this would work in any individual in Africa. And what they wound up doing is increasing the frequency of monitoring in the other countries so that they could stop as early as possible if that evidence occurred, and in fact it did in each of the other countries. So we have some ideas of DSMBs, and I just want to say that they are extremely helpful, and certainly if you have a blinded trial, phase 3 trial, they're compulsory, I think, and NIH policy requires it, and I think many other institutions would as well. But not all trials need the DSMB. There are many other ways to protect the patients. And you can read about these policies from various institutes. NIH, just to give you some general guidelines for when you do need one of these bodies. All trials need a safety monitoring plan, a plan for how they're going to make sure the data in the trial are conducted as intended and have integrity, and also to protect the safety. But as soon as you have multi-center phase 3 trials, that an independent DSMB has been required since 1979. It's also required that the IRBs are notified of all DSMB recommendations. And I say beyond that, the policies start to vary between institutes. I used to work at NIAID, and I know that as soon as a trial was blinded and randomized, it had a DSMB. But that is not true across all institutes. So it would be even phase 2 at NIAID. So here's some links that you can go to read about these guidances. The FDA, I actually think, is a very informative guidance, and would recommend that reading if you wanted to as well. It's pretty comprehensive if you're interested in thinking about the best practices in forming these committees. I would say that there is no policy of the FDA on what you should do, or there's no requirements of what is done, but simply recommendations. Because they're not governing, say, pharmaceutical firms or other, but in terms of how they do these DSMBs, but they will make recommendations on best practice. Same thing for how to do a statistical analysis, how to monitor your trial. The FDA has a lot of guidance documents. If you haven't found those, I'd look online, very useful. I did want to point out also, for those of you who are at NIH, it was interesting that there's quite a while sort of understanding that The centers and institutes differ in terms of how they do DSMBs, and some of them are a lot more open to the idea of the study sponsor being a participant, a heavy participant in the DSMB and the executive session, and some complaints have lodged from investigators about an interference or a conflict of interest. And so the Health and Human Services Office of Inspector General took a couple of years, I think 2009, 2010, and reviewed all the DSMBs across the institutes and centers, interviewed the investigators, DSMB members, the sponsor, and just asked sort of a set of questions to try to evaluate whether these DSMBs were conducted in a way that met NIH's own policy, and if there are any challenges to the way things are currently being done in terms of to the DSMB effectiveness. And this makes for good reading. It's a brief report, but you can see there were recommendations, but there was sort of a back and forth between NIH officials and the Health and Human Services in terms of what recommendations or sort of assessments of what was needed and not needed, particularly on the issue of that independence actually is the real sticking point, I think, between some of the institutes and those outside of the NIH. But the main findings were that the NIH DSMBs did meet the guidance. Overwhelmingly across all those interviewed, there was a few dissenting opinions, but quite a number that said across the investigators through the members that the policies were being met. But there were complaints, particularly from certain investigators, about participation of study staff in the closed meetings was not in all circumstances seen to be helpful, but in other circumstances it was pointed out that it was helpful because they were the experts in certain rare diseases. They were the study staff that had think about these issues day and night. So I think maybe one size does not fit all, but that is still room for debate in how we do these things. I think another issue was that we do these reports as masked, and there wasn't really good understanding amongst all members of the DSMB themselves that were interviewed that they had the right to ask for the unblinding codes. And so there was a recommendation to avoid that confusion, and certainly to make sure this was an accepted policy, that it state in the policies in all centers that the DSMB has the right to see unmasked data, to make sure that's absolutely clear. That was historically seen as a challenge. Some DSMB members had no idea. And then ultimately, what's really interesting about this field is it's incredibly complex. So when you see on someone's resume that they were the one on the CAST trial, and you're doing a cardiovascular trial, you're going to want those same people on your DSMB. And so you have these same experts serving on the HIV trials or the cardiovascular trials, and it's an aging population of experts when you see them talk, and there's a lot of concern that this expertise is going to disappear and that we don't have enough people who are new that are engaging in this activity. And so there's active efforts across NIH and other sponsored trial settings to get new people into this endeavor, which, as you can imagine, is quite complex. So the NIH, it was recommended they should direct the ICs, the institutes and centers, to at least articulate the circumstances in which the institutes and center staff can participate in the DSMB. So at least everyone knows what to expect, and at least in those circumstances understand if a particular IC member is not abiding by their own restrictions. And so that was seen as agreed. Also, it was unilaterally agreed that they would make it more clear that all DSMBs that the policies specifically say that the DSMBs have access to unmasked data. That was an easy fix. And that they're all working on ways to identify and recruit new members. So with this sort of negotiated set of recommendations, the NIH agreed. Okay, so sort of talking about earlier that it's not always needed to have an independent body. You can imagine it's quite a complex and expensive endeavor to get a group of experts like this and fly them around the world to have these meetings. They are sort of universally accepted as important and needed when you have large randomized trials with mortality or major morbidity, certainly. If the trial, for whatever reasons, has implications for public health, you're going to want an independent body to help you through the conducting of this trial. If there's a serious toxicity that's thought to be a risk, or potentially, for whatever reasons, other kinds of high-risk treatments, then people generally, even if it's not blinded, might think about having a DSMB as an independent advisory board to help make decisions about what might be an ethically complex situation. And generally, if there's a vulnerable population, whether it be children or pregnant women or however you might define vulnerable, folks with Alzheimer's or dementia, these would be populations that would need extra protections. When they're not needed, typically if you don't have any of those ethical, tricky, or seen as difficult situations, but maybe run-of-the-mill, allergy medicines, and they're so not anything unusual in terms of risks being taken, single-arm trials, not blinded. There's really no reason to have an independent body if everyone can see the totality of evidence as people accrue. Short-term trials, it's almost impossible to get a DSMB together if a trial is going to be quite quick in terms of accrual and endpoints. And so there's no point, it can't stop early. So what is the DSMB going to do? And certainly, I think when you have that unblinded data with no ethical reason to be extra concerned, there's really no need for a DSMB. And there are disadvantages. So a lot of people just want to be better safe than sorry, but it increases the complexity of the trial management. Once you say you're going to do these DSMB meetings, they have to happen. It increases costs. And so I think the thought is that you should, and to think we have a sort of a small set of individuals who can serve on these boards that have expertise in your area. Don't use them up in a situation where they're not really needed, and keep them fresh and available for situations where you really do need them. So I think that's another important reason, especially in smaller fields, to think about whether you don't have one if you don't think you need one. Because you have many other forms of monitoring. You can have your own local safety monitors, perhaps some kind of partial, if you're not going to be monitoring efficacy, but you want extra experts to monitor safety, then you can recruit those form a specific safety monitoring board without all the other extra goals that a DSMB typically has. You have the IRB, the Institutional Review Board, that's going to look at the human subjects at your institution. And the protocol team themselves should be constantly reviewing how their study is going and the safety of the patients in their study. So, as promised, it's going to go from those big concepts to some just nuts and bolts of example tables or shells of things that I've seen in monitoring reports. Many of you, if you're familiar with clinical research, know about the consort diagram. I think it's useful to see it in the DSMB report, because then you can see how many people are being screened, approached, and how many of those are accepting and accepting and then failing the eligibility criteria later for other reasons. It gives you an idea of feasibility and representation, and then also a great concise way to look at things like, of those that should have been analyzed, what percent were withdrawn, lost to follow-up, that discontinued early, or completed. All of those things, I think, are very useful and quite concisely summarized in a consort diagram. Going back to validity, and are you doing the protocol that you intended? Are you going to have interpretable results? You definitely want to look at missed visits. You want to look at them by visit. You want to look at the baseline. Certainly, if they're not even getting through the baseline, that's a concern. And you want to see how many are completing the last visit. Overdue forms, that might give you an idea of whether or not your protocol is a little too difficult in terms of the schedule. People are willing to do it but can't quite make the timelines. It's useful information. It might also be very fixable. Baseline characteristics, are these the people you intended to enroll? Are they balanced by arm? If randomization worked, these things should be balanced by arm, but randomization doesn't always work, and sometimes it's been discovered in meetings like this by DSMB. Serious adverse events. Now, in many ways, that's a no-brainer. Obviously, a DSMB is going to look at that. But I think what's important is how you present it to the DSMB. If you have 4,000 people in the trial and you're going to present line listings of every sniffle, and headache, they'll never find the issues of concern. And that is a concern. And so you want to be complete. Maybe you have appendices of all the events in case somebody asks a question about a particular one. But you're going to want to think about how to summarize this so the necessary trends can become apparent if there are any. And then you want appropriate detail sort of at the fingertips of start and stop dates. How long did it take for these AEs to resolve? Did they resolve? People often ask, what is the relationship that they think of this adverse event? If something happens to a person on the trial, they fell down the stairs, whatever it is, it has to be reported if there's an injury to a patient in your trial. And sometimes it's quite obvious that maybe it is obvious, maybe it's not. Maybe they fell down the stairs because they were dizzy and it had to do with the treatment. And so essentially you can ask for the expert opinion. The investigators often will list what they think is whether it's probably related, possibly related, definitely related, or unrelated. But ultimately you want it presented to the DSMB so they can make their own opinions. With the opinion of the investigator who's an expert is it's useful to have, but it should also just be considered and not thought of as absolute. So you also then want to start to summarize these maybe by body system. If these can happen multiple times, perhaps you count the number of individuals who've had at least one event, the timing of the first event, the seriousness of the worst event, things like that. The fundamental concept here is that the summaries should be able to easily present, allow trends to just arise to the top. So something that's visually easy to look at, and in this particular one, there's not a lot of interesting data here, but you might think about as a percent of the total number who were treated, how many had any infection or sinusitis, or so you might start with the sum category and then the individual categories of those with at least a 10% rate, and then list them both as overall and then by worst grade. And oftentimes the DSMB, if they don't see something that they want to see, they'll ask for it. And so in addition, of course, if this is part of the plan, you would be looking at efficacy generally for long trials or risky trials or trials where people might stand to have a huge benefit. You might think about interim monitoring. And so you might look at the percent of treatment success, an area under the curve, a Kaplan-Meier curve, and then you would have your official boundaries if you were doing efficacy or futility monitoring. And you would just simply look at the boundary and where you were relative to the boundary and probably the whole history. So you can kind of say how the data are trending. The basic concepts are you want to make it as easy as possible for the DSMB to do their job. So the report should be provided to the DSMB in enough time for the review to happen. They should be able to look at it before the meeting and think about it. Don't email it to them the night before. The report should include a concise summary of the trial itself. Who knows how many boards these DSMB members are on? Typically they serve on multiple because of that expertise that they have. So include that schema, that one-page summary that you have in your protocol telling you the basic endpoints, the number to enroll, the primary objective, the terse analysis plan. You should have an executive summary that processes the main points of what the say for the open report in any case, where you are in accrual and how many new AEs since the last meeting, things like that. And the tables and figures should be concise, easy to understand, and of course accurate. Hopefully I've given you some information today that you can appreciate. The DSMBs, they do have a very difficult job ahead of them and a very important job. They have to balance individual ethics and collective ethics, risk-benefit. They have to think about statistically technical things in terms of being able to interpret interim monitoring efficacy boundaries and inflations of false positive rate. Sometimes they need to consider futility and the balance of what can be gained if you continue versus not continuing. And most importantly, something that sometimes gets lost, which is the decisions are not purely statistical. In general, they are always taking into account much more than a simple boundary or one statistic, and are often very complex in terms of how many different aspects you might consider in terms of overall if the trial should continue as planned or some alteration. And finally, I always like to emphasize, because sometimes investigators, I think, just want this weight of decision-making on somebody else's shoulders. But the DSMB is there to offer advice and guidance, but it is the investigator and the sponsor ultimately who bear the responsibility of stopping or going. And, here are some future readings. I'll say, if like those that I mentioned, if you like suspense novels, read The Data Monitoring in Clinical Trials, a Case Studies Approach. First author, Dave Demetz, who's a great storyteller and who's involved in a lot of landmark trials and interesting DSMBs, where, just unexpected swings and curveballs, and does a good job explaining, sort of lessons learned as well as telling these good stories. Really informative book, as well as the one by Susan Ellenberg and this overall reference by Friedman, Furberg, and Demetz. And the other two here at the end are statistical references. If there are any statisticians out there who want to know more about the nuts and bolts of these boundaries and other statistical issues, those are both very good references. So thank you very much. We'll stop at time for a few questions, if you have any. Yes, and I'll try to remind myself to repeat your question, because I know no one else other than the 20 people in this room can hear you. So go ahead.
Do you ever have situations where the DSMB can't reach a consensus if member experts have disagreements among themselves?
Yeah, so the question was, are there ever, if I sort of aware of situations where the DSMB themselves can't come to a consensus? And yeah, that absolutely can happen. Obviously, you think about, you can have a hung jury, you can have a hung DSMB. And, honestly, I think about, a lot of times in the charter, there are rules such as, first of all, if you want to think about how many members is a reasonable amount for there to be a quorum, because it's impossible to make sure that everyone gets to every meeting. So the first challenge is to make sure you have enough people at that meeting to vote. And others then will say that a decision, there might be a majority-minority, and they will report to the sponsor the outcome of the vote, that it wasn't unanimous. So you have the Supreme Court, you have the dissenting opinion. Because this is advice-giving, in some sense, there's no absolute reason other than it would be nice for the sponsor and the investigator if the DSMB could come up with a clear recommendation. But the answer might not be clear. And so the importance is to present the two opinions. Sometimes you can have a member, and I've certainly heard, it's a bit like social engineering. Like, people need to get along. You need to think about, I think, the personalities involved and can they work together in a difficult situation. And it's not that it's difficult because it's a challenging job ahead. And so if you have a very opinionated person who's not ever going to listen to anyone else's opinion, that's not a great person to have on the DSMB. So there have been times when people have essentially been asked to leave, or it's been uncomfortable enough for them that people just resign. They just no, it's not going to work out, and they're replaced because they can't function well as a body. So a lot of times, I know at NIAID there was one person whose job it was to think about how to make the DSMB. She was in the division, essentially the division of AIDS or anything related to infectious disease. There was. At NIAID, they have separate divisions, say one for transplant or allergies, and the division I was in was focused on infectious disease. And she spent, she networked, she knew the people who served on these bodies, and she would kind of create these bodies and think about, that person's not going to get along with that person, which is kind of stupid in some sense. We're scientists, but at the same time, it's like you guys have to work together. That group has to work together, and personalities inevitably will come into play. Yeah.
Is there a waiver or a challenge process where somebody can go back, the investigator can go back and perhaps challenge what they said?
Yeah, so the question was, is there some kind of waiver or challenge process that an investigator can essentially challenge one of the recommendations of the DSMB? And I can say I definitely was in a situation, I can't remember all the specifics. I was on the study team, not the DSMB, and the DSMB made a recommendation about the treatment. I think it had to do with uncomfortable number of AEs. But the investigator presented an opposing opinion. These AEs were very treatable. This disease was very dire. They kind of came up with something that gave the DSMB more information to process the data. And so there was like a series of letters that were brought back and forth. And then ultimately the DSMB, issued a statement they were more comfortable with, that I think the DSMB might have even recommended stopping. And in the end, they didn't. And they gave reasons why. And then everyone was comfortable and they wound on. And that study was positive in the end. And wound up in the New England Journal. So, the DSMB offers advice. I think if there was a DSMB that felt very strongly, and the WHI, I don't know, to think about, that in itself needs a whole lecture. It's hard to just dabble into that example. But I do know that the first DSMB recommended stopping, and the sponsors, whatever the reasons, they didn't want to, and they continued, and they got a whole other body. And it's and so that's the sponsor's prerogative. But they have public opinion, they have the scientific community that at the end will judge all of these actions. And their sponsors' only responsibility ultimately is to listen to the DSMB, but they are the decision makers. So the idea of challenging that, I think the investigators want to make sure the DSMB understands that they don't want to feel like they're ignoring the DSMB's advice. They want to continue together to continue on in the trial. So a lot of times if you're not going to stop as recommended, the investigators like to explain why and get the DSMB on board, and so they can continue in a congenial and agreeable manner.
How is the DSMB financed? Who pays for their expenses?
So the question is about how are these things financed. So the DSMB, I believe there is an honorarium for the meetings that might require travel. Not always, I don't think. So that is, I'm a little bit unclear. I've definitely been in situations where I've received honorariums and situations where I haven't. But so there might be a small honorarium, and certainly all of your expenses in terms of travel. Most people prefer, especially in maybe ethically challenging or complex trials, in-person meetings. So all that travel expenses, it's all part of the grant. The study sponsor covers all of that. And if the DSMB says, we need an extra meeting, and there's 12 of them coming from around the country, the sponsor has to cough up that money. So they have to have reserve funds to be able to cover a DSMB. And so you make a guesstimate like you do, say, if it's a grant-funded study. And potentially, if it's unexpected burden, perhaps a study team could go back to their sponsor, whether it's the NIH or if it's an industry sponsor, and figure out a way to get supplemental funds if necessary. Patient safety is the primary responsibility. Are there any other questions? All right, well, if not, thank you for your attention, and I'll be for a few extra minutes if anyone else wanted to come up offline and ask questions. Thank you very much for your time.